Hi, my name is Ernest. In the next 5 to 10 minutes, I will discuss how enzyme inhibition is used in modern-day cancer treatment research and follow up with some background knowledge to the principles of enzyme inhibition and the central dogma of molecular biology. The aims I have set out for this presentation begin with the introduction of a recent research publication revising the findings of a breast cancer inhibiting molecule. I will then go on into more depth to discuss the theory behind the research in the treatment of cancer and follow up with the theory behind enzyme inhibition and transcription. Synthesis, enzymatic inhibition, and cancer cell growth inhibition of novel delta-lactam-based histone deacetylase inhibitors is the title of the research publication. The research is centered around regulation of gene transcription by inhibition of an enzyme that the central dogma requires. Delta-lactam-based hydroxamic acids, inhibitors of histone deacetylase, have been synthesized via ring closure metathesis of key diene intermediates, followed by conversion to hydroxamic acid analogues. The hydroxamic acids 12A, 12B, and 17C showed potent inhibitory activity in HDAC enzyme assay. The hydroxamic acid 12B exhibited growth inhibitory activity on 5 human tumor cell lines, showing good sensitivity on the breast tumor cell. The diagram illustrates the synthesis of delta-lactam-based hydroxamic acids via ring closure metathesis of key diene intermediates, followed by conversion to hydroxamic acid analogues. Histone deacetylase and histone acetyltransferase are involved in the co-regulation of chromatin remodeling and the functional regulation of gene transcription. HDAC catalyzes deacetylation of amino groups in lysines located near the N-terminal of core histone proteins. Abnormal recruitment of HDAC is related to carcinogenesis. Thus, the identification of potent HDAC inhibitors has been considered as a very intriguing approach for the development of cancer chemotherapy. A number of natural and synthetic HDAC inhibitors have shown an anti-proliferative activity on tumor cells. The following diagram displays chromatin deacetylation and transcriptional activation with the binding of HDAC to the N-terminal of the histone protein. Growth inhibitory activities of all the active HDAC inhibitors 12A, 12B, and 17C were evaluated in 6 human tumor cell lines. The propionic hydroxamic acids 12B and 17C exhibited good enzyme inhibitory and cell growth inhibitory activities and showed the most potent growth inhibitory activity to breast tumor cells among 6 human tumor cell lines. By efficiently preparing novel delta-lactam-based HDAC inhibitors using ring closure metathesis, it was possible to evaluate their HDAC inhibitory activities and anti-proliferative effects on human tumor cell lines. Almost all therapeutic drugs are enzyme inhibitors, from old medicine box standards such as aspirin and penicillin to the newest compounds used to treat HIV infection. Enzyme kinetics plays an outstanding role in this effort to produce effective therapeutics, for kinetic studies can quantify the degree that inhibitors inactivate or slow down the targeted enzyme's catalytic rate and describe its potential efficiency as a drug. In addition, many naturally produced toxic substances are also enzyme inhibitors. For example, a peptide found in snake venom became a lead compound in the development of a class of hypertension drugs that inhibits angiotensin-converted enzyme. Enzyme inhibitors alter the activity of an enzyme by decreasing the enzyme's ability to bind substrate, lowering its catalytic turnover, or both. There are two main classes of enzyme inhibitors, reversible and irreversible, that are differentiated by the magnitude of their affinity for enzyme. Reversible enzyme inhibitors bind and dissociate with the enzyme in an equilibrium process. Irreversible inhibitors bind tightly to an enzyme to form an essentially permanent complex. Irreversible inhibitors. Because they form such a tight complex with the enzyme that they permanently remove its catalytic activity, irreversible inhibitors are also termed inactivators. For example, kinetic studies on the male pattern baldness drug finasteride reveal it to be an essentially irreversible inhibitor of the 5A reductase. The enzyme catalyzes one step in the conversion of testosterone to dihydrotestosterone, which is a hormone implicated in hair loss. Reversible inhibitors can be classified as competitive, mixed, or non-competitive inhibitors. If the detailed mechanism of inhibition is known, then the classification can be made by identifying where on the enzyme the inhibitor binds, or the order with which it binds relative to the substrate. Competitive inhibitors compete with substrate for an enzyme's active site, lowering the enzyme's likelihood of binding substrate and slowing the observed reaction velocity. Kinetic studies would show an increase in Km, yet the Vmax is unaffected. This is the signature of a competitive inhibitor. Here is a diagram illustrating competitive inhibition. As you can see, there is one active site for which the substrate and inhibitor competes for. A mixed inhibitor binds to a site on the enzyme and interferes with both apparent substrate affinity and catalytic turnover, thus affecting both the observed Km and Kcat for the enzyme-catalyzed reaction. Mixed inhibitors distort the active site, interfering with the chemistry performed by the enzyme. Therefore, the enzymatic turnover rate is slowed. Kinetic studies would demonstrate an increase in Km and a decrease in Vmax. The diagram illustrates how the inhibitor entines the enzyme binding site and distorts the active site for the substrate. This type of enzyme inhibition may also be referred to as allosteric inhibition. Non-competitive inhibition is a special case of mixed inhibition where the affinity of inhibitor for E and ES is the same. Steady-state experiments performed in the presence of non-competitive inhibitor demonstrate a decrease in Vmax, yet Km is unaffected. The diagram illustrates the presence of an enzyme and enzyme-substrate complex for which the inhibitor has affinity for. Kinetic parameters of enzyme inhibitors can be outlined by producing Michaelis-Menten and Lineweaver-Burke plots, allowing for the determination of Vmax, Km, and Kcat. Using a reaction rate graph, a Michaelis-Menten plot may be derived. The parameters displayed by the plot include Vmax at the highest reaction rate and concentration of Km at half of Vmax. Km is indirectly proportionate to the affinity the substrate has for the enzyme, and Vmax identifies the greatest reaction velocity. The Lineweaver-Burke plot is a double reciprocal plot of the Michaelis-Menten results. It is a linear representation of the kinetic parameters of the enzymatic reaction and makes for an easy comparison of Vmax and Km in the presence of different enzyme inhibitors. Kcat is the catalytic efficiency of an enzyme. With a higher Kcat, the enzyme is more efficient because less enzyme is required. The Kcat for a reaction may be determined by calculating Vmax over enzyme times time. And that brings to the conclusion. Hopefully, watching this presentation, I have presented you with a real-life example of how enzyme inhibition is used in modern research and outlined some of the basic principles in the theory of the research. Thank you for watching.